Okay, so our next speaker up is Jeff Morris. He's from the University of Alabama. He has a really interesting background, both in oceanography as well as having done a postdoc with Richard Lenski, looking at microbial experimental evolution. And he's brought that back, that information back with him, and is growing his lab at the University of Alabama, looking at the interactions between microbes and how they evolve. So, thanks, Jeff.
All right, thanks for inviting me. Okay, so biogeochemical models are our friends, right? That's what all of us biologists tell ourselves. We love these things because they help us kind of take our little niches that we study in our labs and put them in a broader context. They help us communicate the importance of our work to the public, to policymakers who really don't care about the nuts and bolts. They just want to know what the world's going to look like in 100 years, or really 10 years, I guess. And they also generate really awesome 3D graphics like this. I think this came from the Follows lab. This is the Darwin model they were talking about, kind of wrapped around the world here. Okay, so that being said, there are some potential pitfalls to the way these models are put together that really kind of stick in the craw of us evolutionary biologists, right, who like to study real organisms. And one of the things, of course, is that we model everything as functional groups, right? So the tens of thousands of beautiful diatom species out there that get some people interested in biology to begin with are just lumped together in one box that eats silicon, right? So, I mean, maybe that's okay, but maybe we're losing something there. And of course, kind of the focus here is that we don't think about evolution, or at least we assume that evolution isn't going to move these species inside this functional group outside of sort of the constraints that we define the functional group as. Right? We assume evolution is happening, but just kind of moving around somewhere inside that box, and it's picked up by the model in that way. And the thing that I'm going to talk about, though, is that the models almost always assume that all the interactions between species are basically negative, right? So essentially, things compete over nutrients, right? The phytoplankton you find in a place inside a model are determined by competition over nutrients. We have exploitation of species where grazers will exploit the phytoplankton to grow. But basically, we don't think about any of the other sort of rich interactions that ecologists think about, things like mutualisms and commensalisms and things like that. We assume that they're just not going to be that important in terms of the biogeochemistry of the system. Okay, so does this ever matter, right? Is this ever going to be relevant from a sort of a global perspective, or can we just sort of ignore all of this stuff? Obviously, I think they do matter, or I wouldn't be showing you that slide. I'm going to give you two examples to start off from my own work that show places where we would have missed some pretty big things if we hadn't looked at species interactions. Then I'm going to talk about the ways that evolution creates these sort of positive interactions between species in the plankton. And then I'll close with a little look forward at some of the things that we're doing, or in some cases that we sort of accidentally did, that are going to let us look more carefully at how these things evolve in the future. Okay, so first of all, I'll just kind of give you a little bit of background about the work that I've been doing for the last 5 years or so. We've been looking at the effects of ocean acidification on phytoplankton. And of course, most of you that have thought about ocean acidification, you know that we normally pay attention to calcifying organisms, right, which are expected to have some serious problems as the pH of the oceans decrease. But one interesting thing, when you look at calcifying phytoplankton, some of them seem to suffer really badly at future CO2 conditions. You see these coccolithophores are all messed up at 920 parts per million here. But other species seem to not really care at all, right? So, one of the first things we did when we got interested in this is we did a big literature review. We did a few experiments of our own to try and get an idea of the overall sort of phenotypic plasticity that Elisa was telling us about of different phytoplankton cultures and their growth rates when they're growing at different CO2 concentrations. So what this graph is showing you is just a bunch of different functional groups of phytoplankton and how their growth rates respond, the ratio of growth at high CO2 to low CO2, across many, many different experiments. What you can see out of this is most of our sort of big eukaryotic phytoplankton, some of them are somewhat enhanced at elevated CO2 concentrations, but most of them basically don't seem to care, as you might expect. Whereas our little guys over here, including almost all the cyanobacteria we looked at, actually had fairly strongly enhanced growth rates at high CO2, right? They grow faster. With the one exception over here of little Prochlorococcus, right? So Prochlorococcus is the most abundant photosynthetic organism in the world. They have these tiny little half-micron wide cells. They numerically dominate the temperate and tropical open oceans. So these guys are pretty important. And they stand out on this graph as being the only functional group that has a significantly reduced growth rate in general at high CO2, right? So the first thing I'm going to talk to you about is how this impacts their competition with Senecacoccus, which is a very similar kind of cell, but has a different ecological profile and a different chemical role in the ocean. So both Pro and Sen, Prochlorococcus and Senecacoccus, they coexist with each other throughout the world ocean, or at least in the open oceans, right? You're going to find both of these types of cells pretty much everywhere. And models that have looked at how warming is expected to affect the distributions of these organisms have predicted in general that both of them will increase in abundance relative to other types of phytoplankton in the future ocean, right? They have, a better tolerance of high temperatures than the larger phytoplankton do. So we expect, just considering global warming, that they'll both become more abundant. But when we took all that data that I just showed you and added ocean acidification in to a model on top of just warming, what we found is that Senecacoccus, as predicted, increases its abundance in the future ocean. But under this simulation, Prochlorococcus totally dropped out of the model and went extinct, right? So that's kind of a striking thing. We don't actually think that's what's going to happen, but just based on our understanding of their response relative to other things, right, they were no longer capable of competing throughout their worldwide niche with Senecacoccus. Okay, so that's a pretty extreme result. So we decided to actually try and ground truth this a little bit. And the way we did this was by taking cultures of Senecacoccus and Prochlorococcus and mixing them together in the same test tube and making them duke it out over a single limiting nutrient, right? So in this kind of environment, right, there's one limiting nutrient. We expect that one or the other of them will have to win and drive the other one ultimately to extinction, right? And we can measure the fitness, the relative fitness of one organism relative to the other by looking at the changing ratios of the two organisms over time. Okay, so we expected, because of Prochlorococcus's reduced growth rate at high CO2, that when we did these competitions at high CO2, Sen would basically wipe the mat with Pro and it would go extinct. This is totally not what we found. We found, and this is a very unexpected finding, we found actually that the initial ratio determined the outcome of the experiment. So when Prochlorococcus was initially rare, it actually had a fitness advantage relative to Senecacoccus. But when Pro was initially common, it had a lower fitness. And this is a pattern that we call negative frequency dependence of fitness, right? In evolutionary biology, this is very important because this is one of very few ways that organisms sharing a niche can coexist with each other. You can see here that there's a point right around 20% Prochlorococcus where the fitness of these two organisms is equal. And indeed, they would migrate towards this ratio and they would stay there and coexist essentially forever in this experiment. So it's kind of surprising to us. So the other thing I'm going to tell you about is the interaction between Prochlorococcus and this heterotrophic bacterium, Alteromonas. So Pro, anybody that's ever worked with it knows that it's a very finicky organism. It doesn't like to grow in cultures. So what you're looking at here on this Petri dish, so essentially Prochlorococcus was applied over this entire Petri dish, but the only place where you see green growth was where heterotrophic bacteria were applied using a toothpick, right? So they have to have these heterotrophic microbes with them to grow in conditions in culture that resemble the natural environment at all. So they also need this in liquid cultures. And essentially, so what these heterotrophic bacteria are doing is they're removing reactive oxygen species from the environment. Prochlorococcus doesn't have any ability to protect itself from oxygen, despite the fact that it obviously produces it. Okay, so what I didn't tell you when I showed you this graph is that we're looking at cultures of algae, but that's not all that's in there. Almost every one of these experiments is done with cultures that had some assemblage of heterotrophic bacteria living with them, right? And that's certainly the case with Prochlorococcus and also with almost all these other ones as well. So what we found when we looked closer at this, we looked at the genetic response of these Prochlorococcus Alteromonas cultures at both modern and future CO2 conditions. And what we found was that at the high CO2 condition, the genetic response of the heterotrophic bacterium actually changed more than the genetic response of the phytoplankton. And again, this is totally unexpected to us. Nobody really thought that the heterotrophs would care about this. In fact, we weren't even thinking about them at all. And among the things that changed were genes that were crucially related to their ability to help Prochlorococcus survive in the media. So this is catalase. Catalase is an enzyme that breaks down hydrogen peroxide. It's key to the coexistence between these two organisms. It's strongly downregulated under future CO2 conditions. We can also look at other genes that change. We can see that biosynthesis genes in the heterotroph go down, transporters go up, and we interpret this basically is that they're kind of stopping being self-sufficient. They're switching from sort of a helping, mutualistic role to sort of an exploitative role at high CO2, right? So the interaction between these two organisms, which is mutualistic under modern conditions, is like antagonistic under future conditions. But surprisingly, when we look back at those Psittacoccus, Prochlorococcus cultures, and again, I didn't tell you that these were also mixed with the same Alteromonas bacterium. When we look at these, the growth problems that Prochlorococcus has at high CO2 go away when Psittacoccus is around, right? So they no longer seem to have a problem with high CO2 as long as Syn, which ought to be like their competitor, the thing that they really don't like, that's competing with them over stuff, it apparently somehow helps them. It makes up for what Alteromonas is no longer doing. Okay. So the interesting thing here is that non-competitive interactions are sort of dominating these little simple ecosystems, right? So we started these experiments to look at competition over nutrients, basic ecology, and instead we ended up studying interspecies interactions, right? Which is not what we were thinking about initially. So Pro and Syn, they should be strict competitors, but instead they also have these positive interactions in addition to competing over nutrients. And I think critically, our interpretation of Pro's response to high CO2 is totally governed by the particular community context that we measure it in, right? So if we look at pure cultures, we get a very different, a skewed and totally wrong interpretation of how they respond to this important environmental change relative to what we get if we measure it in a more natural, complex community setting. Okay, so let's shift from there to talk a little bit about how interactions like this exist, why they exist in the ocean, right? Because it could be that I just happen to pull out 3 organisms that get along, right? It's just luck, and this isn't like really a thing. But no, I think that this is indicative of the way that organisms in the open ocean actually deal with each other. So why are they so friendly with each other? So any evolutionary biologist will tell you that it's really hard to evolve positive interactions between organisms because of this problem of cheating, okay? Natural selection always will favor cheater organisms that can get the benefits of cooperative behaviors without paying the costs. And it's expected that in sort of normal generic conditions, this will drive cooperative behaviors to extinction. And of course we know there's all these conspicuous examples of host-symbiont interactions that have evolved, but there's very particular ways that this can happen. We need structured environments, right, where you can control which organisms get the benefits of what you're doing and exclude those cheaters, right? And there's ways that these kind of systems do that. And importantly, none of that is possible for phytoplankton. For organisms that live in the relatively unstructured pelagic ocean, we don't have structure, right? We can't tell from one day to the next who's going to be next to us. So none of these mechanisms work. So how then do we get cooperation evolving in the plankton? The way I explain it is going to be an analogy from human market economics, okay? So we can think of biology as a marketplace. Every biological function that an organism can perform has a cost. They yield products or services that also have a value. The value is going to be determined based on the payoff in terms of fitness to you, and also by the supply of whatever it is. So as the function becomes more common, its value goes down, just like products in a marketplace. And when the cost is greater than the value in a community setting, natural selection is going to favor organisms that don't perform that function, okay? because obviously it's not worth it to do it. So another thing that's important to think about here is that there's many biological functions that are leaky, meaning that when an organism performs them, some of the benefit of performing the function inevitably gets out from the cell and leaks into the environment by all sorts of different kinds of mechanisms. Either it's actively secreted from the cell, or it just like accumulates in the environment after the cell dies because the product lasts a long time. So there's this theory that we put out a few years ago now called the Black Queen hypothesis. It basically says that market forces around these leaky functions will tend to favor a coexistence between organisms that perform the function and organisms that don't perform the function, right? So here's a graph looking kind of at a model of that, and what you see is that when the function performers are rare, they have a fitness advantage, and when they're common, they have a fitness disadvantage. So this should be familiar to you, is the same trend that we saw in the Prochlorococcus versus Synechococcus competition experiments. And it predicts that there's some intermediate proportion where the two types can coexist with each other. Okay. So when you're thinking about the plankton, right? So all these different organisms are performing various leaky functions. The leaked stuff kind of accumulates out in the extracellular medium, out in the water, where organisms can pick them up and use them, right? So you get what we have started calling a Black Queen marketplace full of all these different things, right? And it leads to this complex web of interdependency. Everything probably makes some things and exploits some things from the environment. And the thing about this is if you were to pull two of these organisms out and culture them and measure the way that they're interacting, you would come to the conclusion, potentially, that they're mutualists, right? That they're in this sort of friendly, cooperative interaction with each other, but really kind of what you're seeing is just sort of like frozen exploitation, right? They're trying to mooch off of each other. They're just not capable of doing it to completion, right? To like driving the opponent to extinction, right? So they're frozen in this equilibrium state. Okay. So that's probably why these organisms that we're looking at seem to coexist. They seem to share these kinds of things. But it's important to think about the fact that these kinds of mutualisms are fundamentally less stable than the kind of mutualisms we associate with like corals or with bioluminescent squid, because they're not evolved because of the benefit of the partner, right? So in a coral, when the zooxanthellae gets a beneficial mutation, the coral benefits, and vice versa, right? Their selective pathways are synced up. But in this particular case, they're not. It's just like happenstance that they end up in this particular state. So you have to ask the question, what happens when we put another kind of evolutionary pressure into the environment? If these guys start to evolve to a new sort of environmental equilibrium, will it disrupt these kinds of interdependencies in a way that's important for the health of the community or for which species and which functional groups dominate at any given time? All right. So this is the experiment that I have been doing for quite some time now. We've been doing this evolution experiment looking at the ability for important functional groups to adapt to high CO2 to ocean acidification. We call this the long-term phytoplankton evolution experiment. So we've been taking Prochlorococcus, Synechococcus, ehux, and the diatom Thalassiosira oceanica. We've grown them for 500 generations, either at 400 parts per million CO2 or at 800 parts per million CO2, right? So 6 replicate populations of each at each CO2 concentration. And the idea originally when we started this experiment was just to look at the pace of adaptation of these different taxa, which have different sort of baseline responses to CO2, to look at their different adaptation rates to the changed CO2 environment, and to see also if there are any sort of secondary phenotypes that changed as well. What I really wasn't thinking too much about when we started this was the fact that, well, we also grew them all with Alteromonas in there, and the reason we did that was because we couldn't get Prochlorococcus to survive in the medium without adding these bacteria. We decided to keep everything as constant as possible across all the lines. So we just grew all 4 of these as a defined community containing 1 phytoplankton and 1 bacterium. So now, having seen how important these sort of interactions are, we're kind of happy that we did this because we have now 48 populations of Alteromonas cultures evolved under different regimes with 4 different kinds of phytoplankton, right? So we can separate them, we can mix and match them, we can see how their phenotypes have changed, how their responses to the presence of the phytoplankton have changed. So I was hoping I was going to be able to show you some of the genetic data from the end of this experiment, but the sequencing guys are kind of slow. So it's probably okay because they don't have time anyway. Okay, so yeah, so just real quick, you know, the outstanding questions that we hope to answer from that is does rapid evolution to environmental change alter these black queen relationships that have co-evolved between these organisms? And also, you know, are there specific, like species-specific interactions that evolve between these bacteria and the phytoplankton they're with? Or are they really on different evolutionary trajectories, right? Because that has, like, you know, pretty big difference in terms of like, you know, how we anticipate these community structures to change over the coming decades. All right, so with that, I will just thank the people who have actually done this, and in particular, Elizabeth and Alex, who have suffered through the daily transfers for years of the long-term experiment, and happy to take any questions.
Prochlorococcus and the Synechococcus. I think you had a slide with a bunch of those in there.
Yeah, well, so the bunch of the scenarios were hypothetical scenarios. The reality is we have no idea how Pro and Syn are coexisting. That experiment went in a totally different direction than we were expecting. It's not reactive oxygen species, okay, because that strain of Synechococcus doesn't make catalase. It doesn't have the same kind of growth defects that Pro has in the presence of oxygen, so it might have some other mechanism for protecting them, but we don't know. That's something we need to find out. All the other stuff there, though, I mean, like nitrogen fixation, siderophore production, leaking of amino acids, those are all things we know that other organisms do that sort of form a marketplace.
Jeff, if we come back to your the initial sort of question you posed, how do you begin to bring in facilitation or some sort of commensal relationship into the modeling framework? I was sort of thinking when you mentioned that about the early work on mixotrophy, which proved very difficult to move into models because when you allow something to be able to jump around its trophic mode in any sort of which way, it sort of takes over the world. Right. So how do we put some sort of
I was thinking, I mean, negative frequency dependence could be programmed in, right? I mean, if we understand the slope of that interaction between different taxa, because, I mean, we know from other studies, from tons of studies people have done, there's widespread negative frequency dependence in the microbial world. And it preserves a lot of things as rare members of the community, right? So if we know that slope and we know the intercept and the shape of that relationship, that could be programmed into a model, even not knowing why it exists.
Yes, so sort of what you're saying is you could tune the model using that sweet spot for coexistence.
Exactly.
and how that tracks with
Right.
the environment. Yeah, that would work.
Exactly. That's the way I would do it. Well, like I said, it depends on what the slope of, what the shape of the coexistence is. I mean, it would preserve more diversity in the model, and my guess is that if you have it done right, it'll preserve a lot of rare diversity, right? Which is the way that the ocean actually works. I mean, there's this long tail of rare things that you find if you look at any given sample, right? As to what effect that would have in the model, I mean, I don't know.
When you do that, did you have to subject those organisms to a limiting condition, like nutrient limiting conditions, so they cooperate? If you have everything in abundance, would they still cooperate?
Okay, so that's a good question. So we grow them in relatively dilute culture media, right? So it's the equivalent of F over 50 nutrients, right? So it's about 1/25th your sort of like rich culture collection conditions. It's definitely more nutrients than you would get in the ocean. And we weren't trying specifically to stress them out. We were just trying to keep them at like a maximum cell density similar to the maximums that they can achieve in nature, right? So it's not rich, but it's also not like the watery desert of the Pacific Gyre. So it would be difficult to do that with our methods, right? So because we're using batch cultures, so if you were to use a chemostat type setup, you could really dial them down. And I have no idea how they would respond to that. It would probably be different.
Out of curiosity, for the name of the Black Queen hypothesis?
Where the name of the Black Queen hypothesis comes from. So it comes from the card game Hearts, right? So if you've ever played Hearts, the goal is to get the lowest score. Most of the cards aren't worth any points. All the hearts are worth 1, but the Queen of Spades is worth 13, so as much as all the rest of the cards combined. So your whole shtick is to not get stuck with the Black Queen. So these Black Queen functions are costly, and your neighbors can cheat on you if you do it. So you don't want to have to be the one that gets stuck with it. But inevitably, in every game, somebody has to. So that's
Which is about how it connects to the Red Queen.
Yeah, well, so the connection between the Black Queen and the Red, I could go on, right? So the Red Queen is like an arms race. It builds up complexity and diversity. The Black Queen, we call it a race to the bottom, right? So the idea is to streamline your genome as much as possible. So they work in like a tug of war, like in opposite directions almost.